Tonight's lecture is over Chapter 2, The Chemical Level of Organization, and it begins on page 29 in your textbook. Maintaining the proper assortment and quantity of thousands of different chemicals in your body and monitoring the interactions of these chemicals with one another are two important aspects of homeostasis. The purpose of this chapter is to introduce the language and fundamental concepts of chemistry, discuss how matter is organized, discuss how chemical bonds form and how chemical reactions occur, and compare and contrast organic and inorganic compounds. And the first thing we'll discuss in regards to the purpose of this chapter is how matter is organized. Chemistry is the science of the structure and the interactions of matter. All living and non-living things consist of matter. Matter is anything that occupies space and has mass. Mass is the amount of matter in any object, and that does not change. Weight is the force of gravity acting on matter, and that does not change. When objects are farther from Earth, the pull of gravity is weaker. This is why the weight of an astronaut is close to zero in outer space. Moving on to the next slide. So matter exists in three states: solid, liquid, and gas. Solids such as bones and teeth are compact and have a definite shape and volume. Liquids such as blood plasma have a definite volume and assume the shape of their container. Gases like oxygen and carbon dioxide have neither a definite shape nor volume. And that brings us to elements. All forms of matter, both living and non-living, are made up of a limited number of building blocks called chemical elements. Each element is a substance that cannot be split into simpler substances by ordinary chemical means. Scientists now recognize 118 elements. Of these, 92 occur naturally on the Earth. The rest have been produced from natural elements using particle accelerators or nuclear reactors. Each named element is designated by a chemical symbol, one or two letters of the element's name in English, Latin, or another language. Examples of elements are oxygen, carbon, hydrogen, nitrogen. However, there are 26 different chemical elements that are normally present in your body. The elements listed on your slide, oxygen, carbon, hydrogen, and nitrogen, those are what we call the major elements, and they constitute about 96% of your body's mass. We have 8 others that we call the lesser elements, and they constitute about 3.6% of the body's mass, and those are calcium, phosphorus, potassium, sulfur, sodium, chlorine, magnesium, and iron. An additional 14 elements are called the trace elements, and these are present in very tiny amounts. Together, they account for about 0.4% of the body's mass. And several trace elements have important functions in the body. For example, iodine is needed to make thyroid hormones. The functions of some trace elements are unknown. But on this next slide, we see table 2.1 on page 30, and this summarizes the main chemical elements in the body. So please review this table on your own time. And this next slide is also part of the same table. And this slide is also part of the same table. And this slide is also part of the same table. And this slide here with the pie diagram on it, this is also part of the same table, but now we're on page 31. Moving on to atoms. Each element is made up of atoms. Atoms are the smallest units of matter that retain the properties and characteristics of the element. Atoms are extremely small. 200,000 of the largest atoms would fit into one period at the end of this sentence. On this slide, we see figure 2.1 on page 31. And this shows us two different representations of the structure of an atom. So electrons move about the nucleus, which contains neutrons and protons. Dozens of different subatomic particles compose individual atoms. However, only 3 types of subatomic particles are important for understanding the chemical reactions in the human body, and those are protons, neutrons, and electrons. The dense central core of an atom is its nucleus. Within the nucleus are positively charged protons and uncharged neutrons. The tiny, negatively charged electrons move about in a large space surrounding the nucleus. They do not follow a fixed path or orbit, but instead form a negatively charged cloud, quote unquote, that envelops the nucleus. Even though their exact positions cannot be predicted, specific groups of electrons are most likely to move about within certain regions around the nucleus. These regions are called electron shells, and they may be depicted as simple circles around the nucleus, as we see in letter B, and that's because each electron shell can hold a specific number of electrons. The electron shell model best conveys this aspect of atomic structure. The first electron shell that's nearest the nucleus never holds more than 2 electrons. The second shell holds a maximum of 8 electrons, and the third shell can hold up to 18 electrons. The electron shells fill with electrons in a specific order, beginning with the first shell. And any time an electron shell has its full complement of electrons, we call that valence. Moving on to the next slide, the atomic number of protons in the nucleus of an atom is an atom's atomic number. Atoms of different elements have different atomic numbers because they have different numbers of protons. For example, oxygen has an atomic number of 8 because its nucleus has 8 protons, and sodium has an atomic number of 11 because its nucleus has 11 protons. The mass number of an atom is the sum of its protons and neutrons. Because sodium has 11 protons and 12 neutrons, its mass number is 23. Although all atoms of one element have the same number of protons, they may have different numbers of neutrons and thus different mass numbers. Isotopes are atoms of an element that have different numbers of neutrons and therefore different mass numbers. In a sample of oxygen, for example, most atoms have 8 neutrons and a few have 9 or 10, but all have 8 protons and 8 electrons. Most isotopes are stable, which means their nuclear structure does not change over time. However, some isotopes are called radioactive isotopes because they are unstable and their nuclei decay into a stable configuration. As they decay, these atoms emit radiation, either subatomic particles or packets of energy, and in the process often transform into a different element. For example, the radioactive isotope of carbon, C14, decays into N14, or nitrogen-14. The decay of a radioactive isotope may be as fast as a fraction of a second or as slow as millions of years. The half-life of an isotope is the time required for half of the radioactive atoms in a sample of that isotope to decay into its more stable form. The half-life of C14, which is used to determine the age of organic samples, is about 5,730 years. Moving on to atomic mass, the standard unit for measuring the mass of atoms and their subatomic particles is a Dalton. A neutron has a mass of about 1.008 daltons, and a proton has a mass of about 1.007 daltons. The mass of an electron is about 0.0005 daltons. This is almost 2,000 times smaller than the mass of a neutron or proton. The atomic mass of an element is the average mass of all of its naturally occurring isotopes. Typically, the atomic mass of an element is close to the mass number of its most abundant isotope. And that brings us to figure 2.2 on page 32. And we see the atomic structures of several stable atoms. The atoms of different elements have different atomic numbers because they have different numbers of protons. Moving on to ions, molecules, and compounds. As we discussed, atoms of the same element have the same number of protons. The atoms of each element have a characteristic way of losing, gaining, or sharing their electrons when interacting with other atoms to achieve stability. The way electrons behave enables atoms in your body to exist in electrically charged forms called ions or join with each other into complex combinations called molecules. If an atom either gives up or gains electrons, it becomes an ion. An ion is a particle that has a positive or negative charge because it has an unequal amount of protons and electrons. Ionization is the process of giving up or gaining electrons. An ion of an atom is symbolized by writing its chemical symbol followed by the number of its positive or negative charges. Thus, Ca2+, calcium ion, stands for a calcium ion that has 2 positive charges because it has lost 2 electrons. When 2 or more atoms share electrons, the resulting combination is a molecule. A molecular formula indicates the elements and the number of atoms in each element that make up the molecule. A molecule may consist of atoms of the same kind, such as an oxygen molecule. The molecular formula for a molecule of oxygen is O2. The subscript 2 indicates that the molecule contains 2 atoms of oxygen. 2 or more different kinds of atoms may also form a molecule, as in water, which is H2O. In water, on the other hand, a compound is a substance that contains atoms of 2 or more different elements. Most of the atoms in the body are joined into compounds. Water and sodium chloride are compounds. However, a molecule of oxygen is not a compound because it consists of only one element. Moving on to free radicals. A free radical is an atom or group of atoms with an unpaired electron in its outermost shell. A common example is superoxide, which is formed by the addition of an electron to an oxygen molecule. Having an unpaired electron makes a free radical unstable, highly reactive, and destructive to nearby molecules. Free radicals become stable by either giving up their unpaired electron to or taking on an electron from another molecule. In doing so, free radicals may break apart important body molecules. Moving on to chemical bonds. The forces that hold together the atoms of a molecule or compound are called chemical bonds. The likelihood that an atom will form a chemical bond with another atom depends on the number of electrons in its outermost shell, and that's also called the valence shell. I mentioned that earlier. And this brings us to table 2.2 on page 35. And this particular slide shows common cations in the body. And we move on to the next slide. We're on the same page, same table, but now we're looking at common anions in the body. Please review this table on your own time. And that brings us to figure 2.4 on page 34 of your book. So remember earlier on in the lecture, I mentioned that the first electron shell only requires 2 electrons for valence. But when we move into the second electron shell, that shell is going to require 8 electrons for valence. This chemical principle is called the octet rule, and it helps to explain why atoms interact in predictable ways. One atom is more likely to interact with another atom if doing so will leave both with 8 valence electrons. For this to happen, an atom either empties its partially filled valence shell, fills it with donated electrons, or shares electrons with other atoms. The way that valence electrons are distributed determines what kind of chemical bond results. And we will consider 3 types of chemical bonds: ionic bonds, covalent bonds, and hydrogen bonds. And this slide here on figure 2.4 shows us ionic bonds. So remember, when atoms lose or gain one or more valence electron, ions are formed. Positively and negatively charged ions are attracted to one another. In other words, opposites attract. The force of attraction that holds together ions with the opposite charges is called an ionic bond. So in this figure, we see a sodium atom can have a complete octet of electrons in its outermost shell by losing one electron, and a chlorine atom can have a complete octet by gaining one electron. So at letter C, an ionic bond may form between oppositely charged ions. At letter D, in a crystal of sodium chloride, each sodium ion is surrounded by 6 chloride ions. And then also in letters A, B, and C, the red electron indicates the electron that is donated and accepted. So moving on to figure 2.5 on page 36, this figure describes covalent bond formation. When a covalent bond forms, 2 or more atoms share electrons rather than gaining or losing them. Atoms form a covalently bonded molecule by sharing 1, 2, or 3 pairs of valence electrons. The larger the number of electron pairs shared between 2 atoms, the stronger the covalent bond. Covalent bonds may form between atoms of the same element or between atoms of different elements. They are the most common chemical bonds in the body, and the compounds that result from them are most of the body's structures. And moving on to the next slide, we see the continuation of figure 2.5 on page 36. Okay, so taking these last 2 slides into account, the red electrons are shared equally, and we see those in A through D. And if we look at letter E, we see that the electrons are shared unequally. So on letters A through D, some covalent bonds share electrons equally, and in this case, one atom does not attract the shared electrons more strongly than the other atom, and this type of bond is called a nonpolar covalent bond. And then in letter E, we see a polar covalent bond, and that's where the sharing of electrons between 2 atoms is unequal. The nucleus of one atom attracts the shared electrons more strongly than the nucleus of the other atom. And then if we look to the right of the figure, we see simpler ways to represent these molecules. So in the structural formula, each covalent bond is denoted by a straight line in between the chemical symbols for the 2 atoms. And in the molecular formula, the number of atoms in each molecule is notated by subscripts. And that brings us to hydrogen bonds. This is figure 2.6 on page 37. So remember, I was talking about polar covalent bonds when we were discussing the covalent bond on the previous slide. So how that relates to hydrogen bonds is that the polar covalent bonds formed between hydrogen atoms and other atoms can give rise to a third type of chemical bond, and that's the hydrogen bond. A hydrogen bond forms when a hydrogen atom with a partial positive charge attracts the partial negative charge of a neighboring electronegative atom, and that's most often large oxygen or nitrogen atoms. Thus, hydrogen bonds result from the attraction of oppositely charged parts of molecules rather than from sharing electrons as in covalent bonds or the loss or gain of electrons as in ionic bonds. Moving on, the hydrogen bonds that link the neighboring water molecules give considerable cohesion. That's the tendency of like particles to stay together. The cohesion of water molecules creates a very high surface tension, and that's a measure of the difficulty of stretching or breaking the surface of a liquid. At the boundary between water and air, water's surface tension is very high because the water molecules are much more attracted to one another than they are attracted to the molecules in the air. This is readily seen when a spider walks on water or a leaf floats on water. The influence of water's surface tension on the body can be seen in the way it increases the work required for breathing. A thin film of watery fluid coats the air sacs of the lungs, so each inhalation must have enough force to overcome the opposing effect of the surface tension as the air sacs stretch and enlarge when taking in air. Even though single hydrogen bonds are relatively weak, very large molecules may contain thousands of these bonds, and then acting collectively, hydrogen bonds provide considerable strength and stability and help determine the three-dimensional shape of large molecules. Moving on, a chemical reaction occurs when new bonds form or old bonds break between atoms. Chemical reactions are the foundation of all life processes, and as we have seen, the interactions of valence electrons are the basis of all chemical reactions. Consider how hydrogen and oxygen molecules react to form water molecules. The starting substances, 2 hydrogens and 1 oxygen, are known as reactants. The ending substances are 2 molecules of H2O or water, and those are the products. If we direct our attention to figure 2.7 on page 37, the arrow in this figure indicates the direction in which the reaction proceeds. In a chemical reaction, the total mass of the reactants equals the total mass of the products. Thus, the number of atoms of each element is the same before and after the reaction. However, because the atoms are rearranged, the reactants and products have different chemical properties. Through thousands of different chemical reactions, the body's structures are built and functions are carried out. And that is what we call metabolism, and that refers to the sum of all the chemical reactions occurring in the body. Moving on to the next slide, each chemical reaction involves energy changes. Energy is the capacity to do work. 2 principal forms of energy are potential energy, which is stored energy, and kinetic energy, which is energy in motion. For example, the energy stored behind a dam or in a person poised to jump down some steps is potential energy. When the gates of the dam are opened or the person jumps, potential energy is then converted into kinetic energy. Chemical energy is a form of potential energy that is stored in the bonds of compounds and molecules. The total amount of energy present at the beginning and end of a chemical reaction is the same. Although energy can neither be created nor destroyed, it may be converted from one form to another. This principle is known as the law of conservation of energy. Moving on, chemical bonds represent stored chemical energy, and chemical reactions occur when new bonds are formed or old bonds are broken between atoms. The overall reaction may either release energy or absorb energy. Exergonic reactions released more energy than they absorbed, and by contrast, endergonic reactions absorb more energy than they release. Because particles of matter, such as atoms, ions, and molecules, have kinetic energy, they are continuously moving and colliding with one another. A sufficiently forceful collision can disrupt the movement of valence electrons, causing the existing chemical bond to break or a new one to form. The collision energy needed to break the chemical bonds of the reactants is called activation energy of the reaction. This initial energy investment is needed to start a reaction. The reactants must absorb enough energy for their chemical bonds to become unstable and their valence electrons to form new combinations. Moving on to catalysts. As we have seen, chemical reactions occur when chemical bonds break or form other atoms, ions, or molecules collide with one another. Body temperature and the concentrations of molecules in body fluids, however, are far too low for most chemical reactions to occur rapidly enough to maintain life. Raising the temperature and the number of reacting particles of matter in the body could increase the frequency of collisions and thus increase the rate of chemical reactions, but doing so could also damage or kill the body's cells. So substances called catalysts can solve this problem. Catalysts are chemical compounds that speed up the chemical reactions by lowering the activation energy needed for a reaction to occur. The most important catalysts in the body are called enzymes. And if we look at figure 2.9 on page 39, we see the comparison of the energy needed for a chemical reaction to proceed with the catalyst, that's the blue curve, and without a catalyst, and that's the red curve. So the gist of it is that catalysts speed up chemical reactions by lowering the activation energy. So moving on to the next slide, after a chemical reaction takes place, the atoms of the reactants are rearranged to yield products with new chemical properties. The first set of reactions we'll look at is called synthesis, and then we'll look at decomposition, then exchange, then reversible reactions, and then the oxidation-reduction reaction, or the redox reaction. So that brings us to the next slide, and this cartoon is at the bottom of page 39. There's no figure number for it. But when two or more atoms, ions, or molecules combine to form new and larger molecules, the processes are called synthesis reactions. The word synthesis means to put together. So as we see in this cartoon, the formation of a water molecule is an example of a synthesis reaction. And we can also refer to a synthesis reaction as anabolism. And that brings us to decomposition reactions, and this is also on page 39. No figure number for this. So decomposition reactions split up large molecules into smaller atoms, ions, or molecules. And in this reaction, we can see how one methane molecule can be broken down into one carbon atom and two dihydrogen molecules. The decomposition reactions that occur in your body are collectively referred to as catabolism. And that brings us to exchange reactions. So this cartoon is also on page 39 down at the very bottom. No figure number for it. But many reactions in the body are referred to as exchange reactions because they consist of both synthesis and decomposition reactions. And this cartoon shows us an example of an exchange reaction. Notice that the ions in both compounds have quote-unquote switched partners. The hydrogen ion from hydrochloric acid has combined with the bicarbonate ion from sodium bicarbonate, and the sodium ion from sodium bicarbonate has combined with the chloride ion from hydrochloric acid, and that forms carbonic acid and sodium chloride respectively. So moving on to reversible reactions, this cartoon is on page 40. Some chemical reactions proceed in only one direction, in other words, from reactants to products. However, other chemical reactions are reversible. In a reversible reaction, the products can revert to the original reactants. However, it should be pointed out that some reversible reactions only occur under special conditions. In that case, whatever is written above or below the arrows indicates the condition needed for the reaction to occur. In these reactions, AB breaks down into A and B only when water is added, and A and B react to produce AB only when heat is applied. Many reversible reactions in the body require catalysts such as enzymes. Often, different enzymes guide the reactions in opposite directions. Moving on to oxidation-reduction reactions. These reactions are essential to life since they are the reactions that break down food molecules to produce energy. These reactions are concerned with the transfer of electrons between atoms and molecules. Oxidation refers to the loss of electrons. Oxidation refers to the loss of electrons. In the process, the oxidized substance releases energy. And reduction refers to the gain of electrons, and in this process, the reduced substance gains energy. And one way to remember this easily is the acronym OIL-RIG. Oxidation is a loss, reduction is a gain. Also, redox reactions are always parallel. In other words, when one substance is oxidized, another is reduced at the same time. For example, when a food molecule such as glucose is oxidized, the energy produced is used by a cell to carry out its various functions. So what's the difference between inorganic and organic compounds? Well, inorganic compounds usually lack carbon and are structurally simple. Their molecules also have only a few atoms and cannot be used by cells to perform complicated biological functions. Inorganic compounds include water, many salts, acids, and bases. Also, inorganic compounds may have either covalent or ionic bonds. On the other hand, organic compounds always contain carbon, usually contain hydrogen, and always have covalent bonds. And most organic molecules are relatively large. So let's move on to page 41, figure 2.10. Water is the most important and abundant inorganic compound in all living systems. So in a solution, a substance called the solvent dissolves another substance called the solute. Usually, there is more solvent than solute in a solution. For example, when you sweat, it is a dilute solution of water, the solvent, plus small amounts of salts, the solutes. Water is a versatile solvent because its polar covalent bonds, in which electrons are shared unequally, create positive and negative regions. And this cartoon shows how polar water molecules dissolve salts and polar substances. So for instance, when a crystal of sodium chloride is placed in water, the slightly negative end of the water molecule is attracted to the positive sodium ions, and the slightly positive hydrogen portions of the water molecule are attracted to the negative chloride ions. In addition to dissolving sodium chloride, water also causes it to dissociate into separate charged particles. Moving on to the next slide. Water serves as the medium for most chemical reactions in the body and participates as a reactant or product in certain reactions. During digestion, for example, decomposition reactions break down large nutrient molecules into smaller molecules by the addition of water molecules. This type of reaction is called hydrolysis. Hydrolysis reactions enable dietary nutrients to be absorbed into the body. In contrast, when two smaller molecules join to form a larger molecule in what we call a dehydration synthesis reaction, a water molecule is one of the products formed. Moving on to the thermal properties of water. In comparison to most substances, water can absorb or release a relatively large amount of heat with only a modest change to its own temperature. For this reason, water is said to have a high heat capacity. Water also requires a large amount of heat to change from a liquid to a gas. Its heat of vaporization is high. As water evaporates from the surface of the skin, it removes a large quantity of heat, providing an important cooling mechanism. Moving on, water is a major component of mucus and other lubricating fluids throughout the body. Lubrication is especially necessary in the chest and abdomen, in general in places where internal organs touch or slide over one another. It is also needed at joints where bones, ligaments, and tendons rub against one another. Inside the digestive canal, mucus and other watery secretions moisten food, which aids their smooth passage through the digestive system. Moving on to mixtures. A mixture is a combination of elements or compounds that are physically blended together but not bound by chemical bonds. For example, the air you breathe is a mixture of nitrogen, oxygen, argon, and carbon dioxide. And then there are three common liquid mixtures. Those are solutions, colloids, and suspensions. So concerning a solution, once mixed together, solutes in the solution remain evenly dispersed among the solvent molecules. A colloid differs from a solution mainly because of the size of its particles. So where solutions are often transparent, a colloid is often translucent or opaque. So solutes in both solutions and colloids do not settle out and accumulate on the bottom of the container. However, in a suspension, the suspended material may mix with the liquid or suspending medium for some time, but eventually will settle out. Blood is an example of a suspension. So when blood is freshly drawn from the body, it has an even reddish color, and after blood sits for a while in a test tube, red blood cells settle out of the suspension and drift to the bottom of the tube. And this brings us to table 2.3 on page 42, and this talks about percentage and molarity. So please review this table on your own time. So moving on to the next slide, acids, bases, and salts. This cartoon is on page 42, figure 2.11. When organic acids, bases, or salts dissolve in water, they dissociate. That is, they separate into ions and become surrounded by water molecules. An acid is a substance that dissociates into one or more hydrogen ions and one or more anions. Because a hydrogen ion is a single proton with one positive charge, an acid is also referred to as a proton donor. In contrast, a base removes hydrogen ions from a solution and therefore is a proton acceptor. Many bases dissociate into one or more hydroxide ions and one or more cations. And that brings us to figure 2.12 on page 43. So concerning pH, to ensure homeostasis, intracellular and extracellular fluids must contain almost balanced quantities of acids and bases. The more hydrogen ions dissolved in a solution, the more acidic the solution, and the more hydroxide ions, the more basic the solution. The chemical reactions that take place in the body are very sensitive to even small changes in the acidity or alkalinity of body fluids. Any departure from the narrow limits of normal hydrogen ion and hydroxide ion concentrations greatly disrupts body functions. A solution's acidity or alkalinity is expressed on the pH scale, which extends from 0 to 14. The scale is based on the concentration of hydrogen ions in moles per liter. A pH of 7 means a solution contains 1/10,000,000th of a mole of hydrogen ions per liter. So the midpoint of the pH scale is 7, and that's where the concentrations of hydrogen ions and hydroxide ions are equal. A substance with a pH of 7, such as pure water, is neutral. So, a solution that has more hydrogen ions than hydroxide ions is considered an acidic solution, and that will have a pH below 7. And on the other hand, a solution that has more hydroxide ions than hydrogen ions is considered a basic solution, and that will have a pH above 7. And that brings us to page 44, table 2.4, and this shows the pH values of selected substances. So I will leave you to review this table on your own time. Moving on to the next slide. And moving on to the next slide. So this again is page 44, table 2.4. Please review this on your own time. And that brings us to buffer systems. Even though strong acids and bases are continually taken into and formed by the body, the pH of fluids inside and outside the cells remains almost constant. One important reason is the presence of buffer systems, which function to convert strong acids or strong bases into weak acids or weak bases. Strong acids, or bases for that matter, ionize easily and contribute many hydrogen ions or hydroxide ions to a solution. Therefore, They can change pH drastically, which can disrupt the body's metabolism. Weak acids or bases do not ionize as much and contribute fewer hydrogen ions or hydroxide ions. Hence, they have less of an effect on the pH. The chemical compounds that convert strong acids or bases into weak ones are called buffers. They do so by adding or removing hydrogen ions. And the chemical reaction we see on this slide is at the bottom of page 43. There's no figure number for it. One important buffer system in the body is the carbonic acid bicarbonate buffer system. Carbonic acid can act as a weak acid, and the bicarbonate ion can act as a weak base. Hence, this buffer system can compensate for either an excess or a shortage of hydrogen ions. So moving on to the next slide, many organic molecules are relatively large and have unique characteristics that allow them to carry out complex functions. And important categories of organic compounds include carbohydrates, lipids, proteins, nucleic acid, and adenosine triphosphate. Carbon has several properties that makes it particularly useful to living organisms. For one thing, it can form bonds with 1 to thousands of other carbon atoms to produce large molecules that can have many different shapes. Due to this property of carbon, the body can have many different organic compounds, each of which has a unique structure and function. Moreover, the large size of most carbon-containing molecules and the fact that some do not dissolve easily in water make them useful for building body structures. Furthermore, organic compounds are usually held together by covalent bonds. Carbon has 4 electrons in its outermost shell, so it can bond covalently with a variety of atoms, including other carbon atoms, to form rings and straight or branched chains. Other elements that most often bond with carbon in organic compounds are hydrogen, oxygen, and nitrogen. Sulfur and phosphorus are also present in organic compounds. And that brings us to page 45, table 2.5, and it shows the major functional groups of organic compounds. So please review this table on your own time. And this slide is also part of the same table, same page. And this slide is also part of the same table and same page. So moving on to carbohydrates. Carbohydrates include sugars, glycogen, starches, and cellulose. Even though they are a large and diverse group of organic compounds and have several functions, carbohydrates represent only 2 to 3% of your total body mass. Plus, in humans and animals, carbohydrates function mainly as a source of chemical energy for generating the ATP needed to drive metabolic reactions. Some carbohydrates are structural. One example is deoxyribose, and this is a sugar that is the building block of DNA. And that brings us to figure 2.13 on page 45. And this shows alternative ways to write the structural formula for glucose. So in standard shorthand, carbon atoms are understood to be at locations where the 2 bond lines intersect. And this brings us to table 2.6 on page 46. And this shows the major carbohydrate groups. Please review this table on your own time. And this slide is also part of the same table and same page. Please review it on your own time. And that brings us to monosaccharides. This is figure 2.14 on page 46. Monosaccharides and disaccharides are known as simple sugars. The monomers of carbohydrates, monosaccharides, contain from 3 to 7 carbon atoms. They are designated by names ending in OSE with the prefix that indicates the number of carbon atoms. For example, monosaccharides with 3 carbon atoms are called trioses. There are also tetroses, pentoses, hexoses, and heptoses. And that brings us to figure 2.15 on page 47. A disaccharide is a molecule formed from the combination of two monosaccharides by a dehydration synthesis reaction. For example, a disaccharide such as lactose is formed from a dehydration synthesis reaction between galactose and glucose, and maltose is formed by the dehydration synthesis reaction of two glucose monomers. And that brings us to the third major group of carbohydrates, which is the polysaccharides. Each polysaccharide molecule contains tens or hundreds of monosaccharides joined together through dehydration synthesis reactions. Unlike simple sugars, polysaccharides are usually insoluble in water and do not taste sweet. The main polysaccharide in the human body is glycogen, which is made entirely of glucose monomers linked together to one another in branching chains. And that is what we see at figure 2.16 on page 47. And that brings us to another important group of organic compounds in your body, and that is the lipids. Lipids make up 18 to 25% of body mass in lean adults. Like carbohydrates, lipids contain carbon, hydrogen, and oxygen. Unlike carbohydrates, they do not have a 2-to-1 ratio of hydrogen and oxygen. So this table is on page 48. This is table 2.7, and this summarizes the types of lipids in the body. Please study this table on your own time. This slide is part of the same table, same page. And this slide is also part of the same table and same page. And that brings us to fatty acids. This figure is 2.17 on page 48. Among the simplest lipids are the fatty acids. Fatty acids are used to synthesize triglycerides and phospholipids. Fatty acids can also be catabolized to generate adenosine triphosphate. A fatty acid consists of a carboxyl group and a hydrocarbon chain. Fatty acids can be either saturated or unsaturated. A saturated fatty acid contains only single covalent bonds between the carbon atoms of the hydrocarbon chain. And an unsaturated fatty acid contains one or more double covalent bonds between the carbon atoms of the hydrocarbon chain. And this slide is part of the same figure, same page. So the most plentiful lipids in your body and in your diet are the triglycerides. A triglyceride consists of two types of building blocks: a single glycerol molecule and 3 fatty acid molecules. A 3-carbon glycerol molecule forms the backbone of a triglyceride. 3 fatty acids are attached by dehydration synthesis reactions, one to each carbon of the glycerol backbone. The chemical bond formed where each water molecule is removed is an ester linkage. The reverse reaction, hydrolysis, breaks down a single molecule of a triglyceride into 3 fatty acids and a glycerol. So moving on to phospholipids. Like triglycerides, phospholipids have a glycerol backbone and 2 fatty acid chains that are attached to the first 2 carbons. In the third position, however, a phosphate group links a small charged group that usually contains nitrogen to the backbone. So moving on to figure 2.19 on page 51, the structure of steroids differs considerably from that of triglycerides. As this figure points out, steroids have 4 rings of carbon atoms. Cholesterol, which is synthesized in the liver, is the starting material for the synthesis of other steroids in the body. Moving on, proteins are large molecules that contain carbon, hydrogen, oxygen, and nitrogen. Some proteins also may contain sulfur. Proteins are much more complex in structure than carbohydrates or lipids, And proteins have many roles in the body, and they are also largely responsible for the structure of body tissues. Enzymes are proteins that speed up most biochemical reactions. Other proteins work as quote-unquote motors to drive muscle contraction. Antibodies are proteins that defend against invading microbes. And some hormones that regulate homeostasis are proteins. And that brings us to Table 2.8 on page 51, and this table summarizes the functions of proteins. So please review this table on your own time. And this slide is also part of the same table and on the same page. And this brings us to figure 2.20 on page 52. So the monomers of proteins are amino acids. Each of the 20 amino acids has a hydrogen atom and 3 important functional groups that are attached to a central carbon atom. And that brings us to figure 2.21 on page 52. Amino acids are the monomers that are used to build proteins. The formation of a peptide bond between 2 amino acids during dehydration synthesis is outlined in this cartoon. In this example, glycine is joined to alanine, forming a dipeptide. Breaking a peptide bond occurs via hydrolysis. And as you can also see, this is a reversible reaction. Moving on to figure 2.22 on page 54. Proteins exhibit 4 levels of structural organization. The primary structure is one-dimensional, and the unique sequence of amino acids that are linked by covalent peptide bonds form what we call a polypeptide chain. A protein's primary structure is genetically determined, and any changes in a protein's amino acid sequences can have serious consequences for body cells. The secondary structure of a protein is two-dimensional and refers to the repeated twisting or folding of neighboring amino acids in the polypeptide chain. 2 common secondary structures are alpha helixes and beta pleated sheets. The secondary structure of a protein is stabilized by hydrogen bonds, which form at regular intervals along the polypeptide backbone. The tertiary structure refers to the 3-dimensional shape of a polypeptide chain. Each protein has a unique tertiary structure that determines how it will function. The tertiary folding pattern may allow amino acids at opposite ends of the chain to be close neighbors. Several types of bonds can contribute to a protein's tertiary structure. The strongest but least common bond is a sulfur-sulfur covalent bond called the disulfide bridge. Also, many weak bonds, such as hydrogen bonds, ionic bonds, and hydrophobic interactions, also help determine the folding pattern. In those proteins that contain more than one polypeptide chain, the arrangement of the individual polypeptide chains relative to one another is the quaternary structure. The bonds that hold polypeptide chains together are similar to those that maintain the tertiary structure. Moving on to enzymes. In living cells, most catalysts are protein molecules called enzymes. Some enzymes consist of 2 parts, a protein portion called an apoenzyme and a non-protein portion called a cofactor. Enzymes are highly specific. Each particular enzyme binds to only specific substrates. Substrates are the reactant molecules on which enzymes act. Of the more than 1,000 known enzymes in your body, each has a characteristic 3-dimensional shape with a specific surface configuration which allows it to recognize and bind to certain substrates. Enzymes are also very efficient. Under optimal conditions, enzymes can catalyze reactions at rates that are from 100 million to 10 billion times more rapid than those of similar reactions occurring without enzymes. Furthermore, enzymes are subject to a variety of cellular controls. The rate of synthesis and their concentration at any given time are under the control of a cell's genes. substances within the cell may either enhance or inhibit the activity of a given enzyme. Many enzymes have both active and inactive forms in cells. And this brings us to figure 2.23 on page 55. And this shows how an enzyme works. So at step 1, the substrate makes contact with the active site on the surface of the enzyme molecule, and this forms a temporary intermediate compound called the enzyme-substrate complex. In this reaction, the two substrate molecules are sucrose and water. At step 2, the substrate molecules are formed by the rearrangement of existing atoms, the breakdown of the substrate molecule, or the combination of several substrate molecules into the products of the reaction. Here the products are two monosaccharides, glucose and fructose. And then at step 3, after the reaction is completed and the reaction products move away from the enzyme, the unchanged enzyme is free to attach to another substrate. And this slide is part of the same figure, same page, and this shows a molecular model of enzyme and substrate uncombined on the left, and then enzyme and substrate combined on the right. And that brings us to nucleic acids. Nucleic acids get their name because they were the first discovered in the nuclei of cells. And they are huge organic molecules that contain carbon, hydrogen, oxygen, nitrogen, and phosphorus. Nucleic acids are found in two varieties. The first is deoxyribonucleic acid, and that forms the inherited genetic material inside each human cell. In humans, each gene is a segment of a DNA molecule. Our genes determine the traits that we inherit, and by controlling protein synthesis, they regulate most of the activities that take place in the body's cells throughout our lives. Ribonucleic acid, or RNA, is a second type of nucleic acid, and this relays instructions from the genes to guide each of the cell's synthesis of proteins from amino acids. And this cartoon is figure 2.24 on page 56. And this shows the components of a nucleotide. Nucleotides are the repeating units of nucleic acids. Each nucleotide consists of a nitrogenous base, a pentose sugar, and a phosphate group. And that brings us to figure 2.25 on page 57. And this shows a DNA molecule. DNA is arranged in a double helix. The paired bases project toward the center of the double helix. The structure of the DNA helix is stabilized by hydrogen bonds. You can see that in the dotted lines. And that brings us to page 58, table 2.9, for a comparison of DNA and RNA. Please review this table on your own time. Moving on to ATP. ATP is the energy currency of living systems. ATP transfers the energy liberated in exergonic catabolic reactions to power cellular activities that require energy, in other words, endergonic reactions. Among these cellular activities are muscular contractions, movement of chromosomes during cell division, movement of structures within cells, transport of substances across cell membranes, and the synthesis of larger molecules from smaller ones. As its name implies, ATP consists of 3 phosphate groups attached to an adenosine. And if we look at figure 2.26 on page 59, we can see the structure of ATP and ADP. The squiggles indicate the 2 phosphate bonds that can be used to transfer energy. Energy transfer typically involves hydrolysis of the last phosphate bond of ATP. Moving on to the next slide, where does the cell get the energy required to produce ATP? The energy needed to attach a phosphate group to ADP is supplied mainly by the catabolism of glucose in a process called cellular respiration. Cellular respiration has 2 phases, anaerobic and aerobic. In the anaerobic phase, a series of reactions that do not require oxygen, glucose is partially broken down by a series of catabolic reactions into pyruvic acid. Each glucose molecule that is converted into pyruvic acid yields 2 molecules of ATP. Concerning the aerobic phase, in the presence of oxygen, glucose is completely broken down into carbon dioxide and water. These reactions generate heat and 30 to 32 ATP molecules. And that concludes tonight's lecture.